All right, so we are on to lesson 2. That's not what I wanted to do. So still talking about DNA. This is what I wanted to do. You should have today turned in your 12.1 homework, and then we're going to discuss the structure of DNA today and look at the components. How do we know um, what DNA is made of and how it's put together? Okay. So components of DNA. DNA, the N and A stand for nucleic acids. So if you think way back to the beginning of the year, nucleic acids was one of our 4 types of molecules that we discussed. We were talking about carbs and lipids and proteins and nucleic acids. So nucleic acids are a polymer, right? They're a chain made up of these building blocks, these Legos, called nucleotides. Nucleic acids are very long, slightly acidic molecules. You should probably have picked up on the acidic part because they're called acids. Okay, and so a chain of nucleotides makes up the nucleic acid. A nucleic acid is made up of, or the nucleotides, either way, are made up of a sugar. In this case, the sugar is called deoxyribose. We have a phosphate group, and then we have a nitrogenous base. Those are the 3 parts to a nucleotide or to a nucleic acid. Okay. They are specifically a 5-carbon sugar. They make this kind of hexagon structure with 5 carbons to them. So we're going to look at that here in just a second. Okay, so all 3 of these are pictures of a nucleotide. So we're going to actually start over here. They're just different models. Okay, so this is kind of the very basic 5-carbon sugar here. I think I said it was a hexagon. It's a pentagon. I'm so smart. Okay, so we have a 5-carbon sugar, and on one side it attaches to a phosphate group, and then on another side it attaches to the nitrogen base. Okay, we'll talk about the nitrogenous bases here in a few minutes, the fact that there are more than one and they have different structures to them a little bit. So here you can see the carbons are all labeled. This one is a little bit better picture of it. So we number the carbons. So in this model, the blue is representing, is kind of highlighting the sugar molecule, the deoxyribose. And we number the carbons. There's a carbon at each one of these corners. So we start here to the right of the O, and that is the number 1 carbon. We actually call them 1'. It's an apostrophe, but we say prime. So you have 1', 2', 3', 4', and then off of the 4' sticking up is a 5' sugar. And that's what we attach to the phosphate. And then the 1' carbon is where we attach the nitrogen base. And the 3' is where we would actually attach to stick them together. Okay, so the phosphate is going to attach to the sugar, which would then attach to the next phosphate. So we can make this long chain. Okay. There are 4 nitrogenous bases that we find in DNA. They have names, but we're just going to call them by their letters. If A, T, G, and C. A is adenine, T is thymine, G is guanine, and C is cytosine, if it matters. But they're probably listed out in your book, so you can see those if you need the full names of them. They are joined together by covalent bonds. So where we join the phosphate to the next base, we have a covalent bond. So it's a pretty strong bond. So those parts don't come apart very easily. So there were several people involved in determining the structure of DNA and how DNA is put together and what it's made of. One of these people is a guy named Edwin Chargaff, and he looked at several different organisms and determined the percentages of these four bases in different organisms. Okay, so the percentage of A, T, G, and C. And what he found was that the percentage of A, adenine, is approximately equal to the percentage of thymine. And the percentage of cytosine is approximately equal to the percentage of guanine. Okay, so you can see it doesn't, I don't think for any of these it's identical, but they're close. They're approximately equal. So he didn't really know the significance of that, but it was kind of an interesting find. Rosalind Franklin was another person involved in solving the structure. Her research, she took an X-ray of DNA. And this is actually a picture of the actual thing. This is the actual X-ray that she took. The pattern shows this X shape, which is characteristic of a double helix, or two strands that are twisted around each other. Okay, so the pattern of the X-ray shows that we have strands that are twisted. The X shape indicates that there's two strands in this picture, and she surmised that probably the nucleotides are in the center. The last people we're going to talk about that are involved in solving the structure are Watson and Crick. And they were trying to create a model of DNA, and they couldn't create one that accurately showed the known properties and the fact that DNA could be responsible for storing and transmitting genetic information until they saw Franklin's X-ray. And that let them create this model that could explain DNA's properties. And so what we know about DNA now is that, in fact, it is two strands that are twisted around each other. Okay, that's what we would call a double helix. So a helix is like a twisted ladder, right? And if it's a double stranded, it has two sides to it, that explains Chargaff's rule a little bit, because you could have an A on one side and a T on the other. So every time you have an A, you have a T. Okay, so that's what we call base pairing, which is really what explains Chargaff's rule, is the base pairing, that A always pairs with T. So if every time you have an A, you have a T, then their percentages would be approximately equal. Okay, and every time you have a C, you have a G, their percentages would be approximately equal. And then they also determined that these two strands are anti-parallel. So they're next to each other, right? They're parallel, but one runs this way and one runs this way. And what we mean by that is the orientation of the sugar. Okay, so on this side, you can see that the sugar is at the top. And on this side, the sugar is at the bottom, right? It's upside down compared to this side. So that's anti-parallel. So we read things from the 5' end to the 3' end. So you would read these two sides oppositely. All right, the last thing that we need to know is about the hydrogen bonding that occurs. So hydrogen bonding occurs in between the bases to hold them together, to hold the two sides together. So in fact, the nitrogenous bases are in the center. And they pair together, right? Then we have that base pairing. And the base pairing is actually due to the number of hydrogen bonds that they each can make. So guanine and cytosine can each make these 3 hydrogen bonds, and then adenine and thymine can each make 2. Okay. So it's very difficult to pair a one that makes three hydrogen bonds with one that would only make two. They don't like to do that because then you have somebody's got this extra bond and it's not very stable. So the hydrogen bonding is going to be important when we start to talk about replication. Hydrogen bonding is not a very strong bond. So that'll be important when we talk about how we can break apart these two sides to make more DNA. All right, so I think that is the end. I think I got all the things. Let me know if I was confusing or didn't explain something very well. And tomorrow you should be working on a lab where you're going to build a model of DNA. So I actually have for you to use a model to put together. I only have one, so you're all going to work together. You can't let Piper do the whole thing. And it'll probably kind of take all of you working together to get all the pieces. They kind of snap together. You have a second day to work on it if it's not working out or something. Otherwise, you'll get to move on to the next lesson. But that's what you're supposed to do in the lab tomorrow is use that model. There are directions. They will generally be pretty good about how you should put the pieces together.